Hi guys, I'm hoping this is about to work. There we go. Right. Okay, welcome back if you are joining me. Hi Ellie. We are going to start with thermoregulation. So, thermoregulation, obviously, we've been talking about different stimuli, and if we're looking at thermoregulation, we have to talk about the fact that when we have an endotherm or an ectotherm, it's about them regulating their body temperature and how they are able to regulate it. So when we look at endotherms, sorry, let me just answer Eva's question. Can you get asked stats test questions on paper one? Yes, you can get asked stats test questions on any of the papers. So just be aware that that could come up. It just depends where they decide to assess it. There isn't a only this paper and not that paper. Okay, so when we look at endotherms, we know we are endotherms. We know that we are able to maintain our body temperature within a very narrow range. So obviously we were talking about homeostasis earlier, and we were talking about the ability to maintain within this normal range. So the body temperature is exactly the same. What we do as endotherms is we use heat produced internally from metabolic processes. And so by using the heat produced internally from these metabolic processes, we are able to maintain our body temperature at the limit, which is about 36.5 to 37.4, something along those lines. Now, we do that mainly by using, you are very welcome, using physiological responses. So I'm going to go for another 2 hours. And I hope I'll manage to get through. I'll try and do as much as I can in those 2 hours. And then, I'll stop there because I need to obviously get myself ready and go collect my girls. So I'm going to go for 2 more hours, and then hopefully that should have covered most of the stuff. So when we talk about endotherms, we're talking about mainly using physiological responses. When we talk about ectotherms have their temperature varying with the environment. Now, I don't know why. I have a feeling that fish is coming up somewhere in this paper of yours, potentially. Now, fish are an ectotherm, and fish obviously live in water, and I'll talk about water a bit later on, but I would say potentially they could ask a question linking to the fact that because water has a high specific heat capacity, that would therefore mean that stable environment, they will be able to survive. No, I'm not available tomorrow because my students are all back to me tomorrow, so I'll be doing my one-to-ones with all of them tomorrow in preparation for the actual exam. So I'm not available tomorrow. This is why I'm trying to do it all today for you as much as I can. Okay, so if they give you a graph, if they're going to give you any type of graph, make sure you are reading the information from the graph. So in this case, we're looking at the ectotherm. We can see here in red, and we can see that as we increase the ambient temperature, we are finding that their body temperature also is increasing. So why is that important? Well, you have to understand that with regards to the ectotherms, if their ambient temperature is affecting the body temperature, that's therefore going to affect their metabolic processes. And so what we tend to find is they will use behavioral responses with some physiological responses, but their metabolic processes may be limited. Now, that's okay because they're ectotherms, they will have a lower metabolic demand. And by having a lower metabolic demand means that there is less need for the transport of substances through. So they could look at it in a case of going back to homeostasis, being whether it's negative feedback or positive feedback. So if we're looking at negative feedback, we're talking about a stimulus which is being detected by those sensory receptors that we talked about earlier. by a coordination center, then coordinating what is going to be the response. The effectors are going to carry out that response, and it will bring us back to the normal. So we are reducing the magnitude of whatever the stimulus may be. So what you have to do, Ella, is take a breath and look at the question. When you say that you get the content, but when the question comes up, you just go blank, it means you haven't found the breadcrumbs in the question that are there to guide you on how to bring that information forward. So any stem of the question, I annotate all over those questions, and I would be writing little notes to myself. If they're saying protein, I'm putting globular protein, tertiary structure. If they're talking about, it is on YouTube live right now, with my screen being displayed. So if you want to jump over and join us. So if they're talking about this, for example, if I said that the temperature is increasing, that is the stimulus. If I then talk about something that's going to decrease the temperature, I know that would have to be negative feedback. So it's about understanding the key parts to it. In this case, if I'm reducing the magnitude of the stimulus, I therefore, reducing that magnitude, it's got to be negative feedback. If I'm going to go on the opposite side of things, if I'm going to intensify the original stimulus, so again, any type of stimulus, I am going to have a sensory receptor. Is it the photoreceptor? Is it the thermoreceptor? Is it the chemoreceptor? Is it the mechanoreceptor? Whatever the example is, and that's where you have to bring the links together. So positive feedback, am I intensifying the original stimulus? So I've got a sensory receptor, I've still got a coordination center, I've still got an effector, but this time I'm pushing the system further in the same direction. So if we were looking at, and this would be more paper 2, but if we were looking at fever, would it be paper 2? Paper 2 or paper, well actually it could be paper 1 because it's just about thermoregulation, but if it was fever potentially linked to the immunity, would be paper 2 or 3. But if I'm looking at a fever, what am I going to do? The stimulus is obviously the increase in temperature. When I've got a fever, I push that further to make it hotter. So I have an increase in my temperature, and then I'm going to make it even hotter, so I'm increasing the temperature further. That is positive feedback. If I'm looking at neuronal communication, if I have my sodium coming in and depolarizing the membrane, when I open my voltage-gated sodium channels, more sodium's flooding in, I'm increasing the amount of sodium. So therefore, I am pushing the system further in the same direction. That is positive feedback. So if it is going to intensify that original stimulus, it's positive. If it's going to reverse that stimulus, it's negative feedback. So it's really about looking where in the question they've told you which way around it's going to be. So if we were looking at thermoregulation then and looking at it in us, we would be looking at, obviously, our blood temperature rises. If our blood temperature rises, because it's the blood, we know that we have got our thermoreceptors in our hypothalamus. They are going to detect that core blood temperature rising. They are then going to activate the cooling response, and that would be activating it by sending any type of, whether that is a nervous communication or whether that's a hormonal communication, depends on the question. And so the hypothalamus is going to, in this case, send nerve impulses, and those nerve impulses are going to cause vasodilation, they're going to cause sweat production, they're going to stimulate for our muscles to relax, causing our hairs to lie flat, and that will therefore help to lower the blood temperature. Now, Khadija, you've asked about why is positive feedback used. If we're looking at it with regards to a fever, why do we push our temperature higher? we are trying to slow down the infection. So we're trying to slow the replication of the pathogen and the spread of the pathogen. So by increasing the temperature, we are going to affect proteins. And those proteins are not just our proteins, they could be the proteins in the pathogen. And therefore, by increasing it to a higher temperature, we can slow their rate of reactions down. So positive feedback is a form of increasing that magnitude, whatever it may be. It did come up a couple of years ago with regards to they used a special, they named a special chemical that was binding to a receptor on a cell, which therefore signaled for that. So it was an application of a novel idea. So yes, it could come up. Okay, so if we're looking at our blood temperature lowering, our hypothalamus is going to start to first detect, then it's going to coordinate, and then it's going to communicate. In this case, it would be nervous communication, and that would increase the body temperature because we'd have our erector muscles causing our hairs to stand up. We trap a layer of air, we insulate the body. When we shiver, we are signaling for our muscles to contract and relax at a faster rate. And if they relax and contract at a faster rate, they're therefore going to generate that heat to warm us back up. And if we do vasoconstriction, we are therefore going to constrict the blood flow, less blood flow to the surface of our skin, less blood flow to the arterioles, less radiation going out. So it's really dependent on what needs to happen, but in this case with thermoregulation, it is very much a negative feedback system. Yes, this is OCR that we are doing now. Okay, so we were talking about thermoregulation and why it's really important. If our body temperature, just if I go back a second, if our body temperature gets too low, then we are getting below our optimum for our enzyme activity. So if we have less thermal energy, we have less kinetic energy, we will have less collisions, therefore the rate of reactions within the cells will be decreased. Our metabolic rate will decline if our body temperature gets too low. If our body temperature gets too high, yes, we're increasing our kinetic energy, we're increasing the rate of reaction, but if we go too high, and be aware of that, have they mentioned that we've gone too high? Because it's only depending on the organism where that range lies. So they did a question on the mole rat once, and they had a different narrow range, and so therefore I would say just be careful that you don't start writing out something that's not in the question. So look for those hints that are there. Vasoconstriction is for warmth, yes. Vasodilation is for cooling you down, Ella, that's correct. Vasoconstriction means I'm pulling the blood away, keeping it at the core. Vasodilation is that I'm allowing it to flow through the arterioles, which are at the surface of the skin. So my face, I can see here, I'm getting really red patches on my face and that. So I've got more dilation occurring, and therefore, and I'm going to get up my arms as well. The dilation is occurring, so I am radiating more heat because I'm talking lots, because I am moving my hands, I'm generating heat. Because for my muscles to contract, they require ATP. For ATP, I therefore need to be doing respiration, and the ATP is going to be hydrolyzed, and when it's hydrolyzed down to ADP, that's going to release energy. So if I'm doing lots of things, I'm going to be generating lots of energy. I need a way of cooling the body back down. So by dilating my blood vessels, I have that the blood flow goes closer to the surface of the skin. Therefore, there is less of pathway for that to go through. So what I would say is make sure you know the connections as to why we need to be talking about that regulation. That's what it's about. And so if we're looking at enzymes and we're talking about The fact that we know for a reaction to occur, these molecules need to collide correctly, they need to have sufficient energy, and that is known as the activation energy. If we include our globular protein, our enzymes, we are therefore going to have a biological catalyst. We are therefore going to allow for temporary bonds to actually be made when they form a enzyme substrate complex. So by having a successful collision, by having an enzyme substrate complex forming, we will therefore lower that activation energy. So if we have the right temperature, the optimum temperature for the enzymes for the rate of the reaction, we can speed up the rate of the reaction. So we know that our enzyme substrate complexes need to be formed. We know that they will put a strain on the bonds within the substrate, and that will therefore result in a lowering of our activation energy. And you should know the difference between whether it's the lock and key and whether it's the induced fit model. So know that we have a globular protein. Because we've got a globular protein, we know that it has a tertiary structure. We know that that can change in the induced fit model, and as it changes its structure, it is going to strengthen that binding and put more strain on the substrate. So although we used to believe in the lock and key model, we now are using the induced fit model because that actually makes more sense with the evidence that we have. Now, if you have a rate of reaction, as you increase substrate concentration, obviously you're going to have a higher collision rate, more enzyme substrate complexes forming, and that will therefore mean that you will get to the maximum rate of the reaction. So the minute you see a plateau, that is where you have reached the maximum rate of the reaction. So be very aware when you are doing questions like this that you are looking at what have they actually given you on your axes, because that's going to be your key to tell you where they want you to go. So I don't know why, but a lot of students say that they know the content, get a question and get confused. And I find that that tends to be because they haven't dissected the question, they haven't dissected the graph, they haven't dissected the table that they've been given. So you need to take time, you need to really critically engage with the content that's been given to you in that paper. So obviously, if my substrate concentration is lower, some of those active sites are free. If I get to the point where I have plateaued, that means that all of the active sites are now being filled, and it has to wait for that to finish its reaction before it can accept another one. And that's why you get a plateau. The other thing to note is whether they've used describe or explain as the command word in the question. So that's the other thing to note when you are, if it's describing, I would be saying as the substrate level increases, the rate of reaction increases up to a point and then it levels off. And if I am given actually a proper graph that has a scale that I can use, I would say between here and here, here and here, and I would explain, describe what's happening. If I'm asked to explain, then it's say the science that's going on for giving you that set of results. Obviously, we're talking about temperature. If I increase the temperature, I'm increasing the kinetic energy. Don't forget about low temperatures. If I have low temperatures, I have less kinetic energy, therefore less frequent collisions, therefore less enzyme substrate complexes forming. So many students forget the other side for some reason, and I don't know why. Obviously, last year they did a question on temperature coefficient. So basically, when you do your Q10, it's about if I increase the rate of reaction by 10 degrees C, what is the increase in that rate of reaction? So if I'm increasing my temperature in 10 degrees, normally the rate will double. Well, you need to make sure that you are looking at whatever results that they have given you. I'm actually showing it on YouTube Live. So if you want to come and look at the actual screen and what I'm doing, it's on my YouTube channel too. So the Q10 that you're looking for would be R2, the highest temperature, divided by the lowest temperature, and therefore you would work out your rate your Q10 is 1.88, which obviously if we did some rounding, that would show us that it doubles. But be careful not to round too quickly. Obviously, with our optimum temperature, the optimum temperature is the point at which the rate is the highest. Now, be so careful. So many students go, ooh, like I have, that's the optimum temperature. Is it the optimum, or is the optimum somewhere in between here? How would I know if that's the optimum temperature? And this is where they'd expect you to use your knowledge and say, okay, if I want to, I've done an investigation and I'm saying that that's the optimum, I want to know if that's truly the optimum value. If so, I would like to redo smaller increments between those temperatures, whatever those temperatures are on your graph, and that would therefore give me an indication as to what is the true optimum. How do you know what temperatures to use? If you were doing the investigation yourself, then you would probably want to go up in, I would say, either tens or five. So 5, 10, 15, 20, 25, 30, 35, 40, 45. Now that's quite a lot of temperatures. And so because that's quite a lot of temperatures, it's like, will you have enough time to do that many temperatures? So actually, do I need to go down to 5? It depends on the investigation. I would say going up in tens is sufficient enough, and you should ideally get 5 values that you want to give. In the question, if they give you a graph, then I would say you read it off whatever numbers they give you in the graph. Obviously, I don't have a scale here. This is just a sketch graph, and so it's following the rough trend of what I'd expect. Now, don't jump to denaturation too quickly. That's another thing that a lot of students struggle with. They jump to denaturation. They talk about the fact that high temperature is going to become denatured. You need to know about the organism, because if we're talking about prokaryotic organisms, thermophilic bacteria, they may have higher temperatures for their optimum, and therefore we can't guarantee it's denatured. So where has the source of the enzyme come from in your investigation? Have they told you it's from a prokaryote? And if it has, don't assume denaturation unless you are seeing a point where it is decreasing. Okay, so that's one of the other things to note. And then if it's not about the temperature and the sensitivity of the temperature, it could be talking about the pH, and so therefore it may be that you're looking at different enzymes and how they are affected. So if you have different enzymes, they are potentially going to have different optimums, and if we go away from the optimum, are we affecting the structure? So we know that the hydrogen ions are going to interact with polar and charged R groups, therefore affecting that tertiary structure as we move away both sides of the optimum. And the key thing about pH is that when we do pH, we are looking at it being a reversible denaturation, and because it's reversible, what we would tend to use in any practical is we would use a buffer solution, and therefore the buffer solution will buffer the solution to keep it at its optimum. If we can't use a buffer solution, ideally we want to measure the pH, and therefore we would use a pH probe on that one. And then I'm going to leave you for 1 second, guys. I do apologise. Give me a second. Sorry guys, I am back. Okay, so if we're looking at inhibitors now, if they're doing anything on inhibition, the key thing for you to know is what have they told you about the inhibitor. So if they're telling you about the inhibitor and the inhibitor is going to be blocking the active site and it blocks it temporarily, that therefore means that it is a competitive inhibitor. It is competing for the active site. If we're looking at a non-competitive inhibitor, that is going to bind to a allosteric site, a site away from the active site, and that is going to bind to it, which changes the tertiary structure. If we change the tertiary structure, we are therefore going to mean that the active site is no longer complementary. And if the active site is no longer complementary, that will then mean that rate of reaction is not going to be able to be sustained. So what you have to know is if you're looking at a graph and they're showing this, you've got the rate of reaction. If there is a fixed volume and we have no inhibition, so no inhibitors, this is our rate of the reaction up to our maximum. So by adding in a competitive inhibitor, a fixed volume of competitive inhibitor, and a fixed volume of a non-competitive inhibitor, we know that that competitive inhibitor is going to decrease this rate. It's going to decrease it, but if we increase the substrate concentration, we can overcome the effect of the competitive inhibitor. And so what you see is the rate of reaction is slower, but it will get up to the point where it reaches that VO2 max. VO2? V max even, because I've got VO2 coming in later. So, if we're looking at a non-competitive inhibitor, we're going to decrease the rate of reaction, and therefore you're never going to reach the V max. So, do you have to pick two temperatures at exactly 10 degrees difference? Yes. If you are calculating your Q10, then they'll either, which they did last year actually, they gave a question where it was like 13 degrees C, 23 degrees C, and a 15 degrees C, or 18 degrees C actually. And so what they're trying to see is, do you actually understand that with a Q10, it must be 10 degrees difference? So that's why it's called the Q10, because it must be 10 degrees difference for you to see whether the rate has doubled or not. So yes, you would need to pick two temperatures that are exactly 10 degrees different. Davia, I'm going to be covering transporting plants after I've covered all the stuff that I've got right now. So it is coming. I've left all the plants to the end, but it is coming. So don't worry, that is there. Another one that they haven't done for a while, but I do feel like could come back in this year, is about end product inhibition. So end product inhibition means that we can actually regulate the rate of a metabolic reaction by preventing the overproduction of the end product. So the end product is going to become the inhibitor of somewhere in the pathway that leads to making the end product. So the product is the inhibitor, and it is going to make sure that we stop producing itself. So it depends where it's going to lie in the pathway that they give you. So for example, lysine is the end product to stop oxaloacetate. So if there's too much lysine being produced, that will stop oxaloacetate. Stop the enzyme that's used to convert oxaloacetate to the intermediate one in my example, if I actually talk about it properly. So this is inhibiting this enzyme. So if I call that enzyme number 1, enzyme number 2, number 3, number 4, number 5, I can then say that lysine is a end product inhibition for enzyme number 1 and enzyme number 4. Now, because we know it's an end product inhibitor, we know that it's going to reversibly inhibit. We don't want it to be permanent, so we know it's going to be reversible, and we know that it's going to be non-competitive. It's going to bind to the allosteric site, because the active site for this one is for oxaloacetate. The active site for 4 is the intermediate 3. So it's inhibiting, and it is a non-competitive inhibitor, and it is a reversible inhibition. Okay, so jumping over then to insects. Now they did a little bit, one question which was on insect circulatory system. So they haven't done gas exchange in insects, so that could come up. Now you should know about your spiracles. These are the openings all the way along the side of the insects. You should know that if they are going to open and close their sphincter, it will increase ventilation. So ventilation is different to gas exchange, which is different to respiration. So there's 3 different keywords you should know. Ventilation, if I'm ventilating, if I open the window, I'm letting fresh air in, I'm letting stale air out. I am ventilating this room. And so by opening and closing the spiracles, by opening and closing these sphincters, that is going to ventilate the tracheal system. If I am thinking about, and we're going to talk about the mammalian one, then you know that we are going to ventilate it by using our intercostal muscles. If the end product builds up, is that when it will? Yes. So Khadija, if the end product, if there's too much end product, we want to stop making the end product until we need it to be made again. So yes, if the end product builds up, that is then going to become a non-competitive inhibitor for the production of itself. And then that is going to only be stopping it for a short period of time until the cell can get what am I trying to say here? Until the cell can use the products that it's made, and then it will stop being the inhibitor. So it's kind of like my printer is printing out too many things that I can't put the printed documents into the envelopes at a fast enough rate. So I'm going to pause my printer for a short amount of time. I'm going to pause the pathway until I can deal with the build-up that I've got, and then once I've dealt with that, then I can let it go again. And so it's that way of controlling and regulating the production of itself. So back to the insects then, and back to the way in which the tracheal system works. So we know that we have got these trachea, which are air-filled branched tubes. So the spiracle is just the opening at the end. Then we have the trachea, and then we know that this is remaining open because of chitinous rings. So the insects have a different polysaccharide that they make, which is going to be used, which is going to keep the trachea open. If we were comparing it with ours, we have our cartilage. Our cartilage is going to keep our trachea open. The chitinous rings are full circles. So they're circular rings, and they ensure that they don't collapse, because the insects require this channel, these branched tubes, to allow for the gas exchange that needs to occur. So they need the delivery of the oxygen up to the cells, and they need the removal of the carbon dioxide. Now obviously it goes all the way to the ends to where the cells are. So these are kind of like these tubes and channels that go all the way to the ends, which are called the tracheoles. And the tracheoles are even more minute elongated cells, which are going to form these many tubes that run in between the cells. Now what are we doing? By having these branches, we're going to increase the surface area, and therefore increase the places, the points where exchange can happen. Okay. So If we get towards the end of the tracheals, there is tracheal fluid. Now, the tracheal fluid is important because the tracheal fluid is needed to prevent dehydration. So just like we are potentially becoming dehydrated, as you can see, I keep having to drink because I'm talking so much. I am losing water vapor when I am breathing, and so therefore the more I do, the more potentially I lose, and I could become dehydrated. So I have to replenish that. So they have tracheal fluid to prevent dehydration too. However, the tracheal fluid can actually limit the penetration of air by diffusion. And so by having these high metabolic activities within the cells, what happens is there's a production of lactic acid. Now, lactic acid building up will actually cause water to move out of the tracheoles by osmosis, and that will therefore expose a larger surface area. So it really depends. If they want to ask a question on this, they will be looking at your understanding of you changing the water potential. If I am producing a solute which is dissolved within my cell, that is therefore going to change the water potential and therefore allow water to move by osmosis. And if we're looking at any larger insects, you should know about this ventilation. Yes, they have the spiracle where they can open and close the spiracle sphincter. However, if they're larger and they have a higher metabolic demand, they're therefore going to need a better ventilation system. And so they may do what's known as muscular pumping. And again, very important, changing the volume and the pressure. Whether we're talking about mammals, whether we're talking about insects, whether we're talking about fish, it's important we understand that it is about us changing volume, which would therefore change the pressure, and that would therefore make that movement of air flow increase. They could have a collapsible enlarged tracheae where they have what's known as an air sac. So they fill the air sac up before they fly. As they're flying, they can then collapse that air sac down, which pushes the air to the cells, allowing for that respiration to still occur. Now, if we switch over then and look at our fish, because I do think fish are coming in, they did already look at the exchange and the adaptations of fish to do with gas exchange. It doesn't mean it's not coming up, but it could come up in a different way. So, as I said before, it could be linked to them being ectotherms. It could be linked to them living in aquatic environments. It really just depends where they want to go with the question. So some of the key features that you should know about. If we're talking about moving water in through the fish, we need to talk about our buccal cavity. And the buccal cavity will lower, increasing the volume, decreasing the pressure. Water moves into the mouth. If we then lift the floor of the buccal cavity back up, that will therefore increase the pressure by decreasing the volume, therefore moving the water over the gills. And if we open the opercular valve at the back, that is going to also change the pressure, and therefore if we're going to move the sides of the fish buccal cavity in further, that is going to then force the water out. And so it's really important to understand that whatever they ask you, if they're asking you about fish, knowing that it is that buccal cavity lowers, increase volume, decrease pressure, rises up, decreases volume, increases pressure, forces it out, and the valve is at the back, which is going to obviously open and close depending on how much is going out. So remember, metabolic demand is your metabolism is talking about all the chemical reactions that are happening. Whenever we talk about the metabolic demand, we do tend to refer to respiration, and therefore the need for respiration to produce the ATP, and the ATP being required for the chemical reactions that are happening. So it's all interlinked. So if we're talking about that higher metabolic demand, we're therefore saying that we're probably needing more ATP. If we need more ATP. Where does that come from? That comes from the respiration. So it's kind of like all of that, depending on if they want to be specific or if we're just saying generically that high metabolic demand. Okay, so if we look at the mammals then, and we talk about mammals, and we know we obviously have our nasal cavity, we know we have our mouth, we know we can breathe air in, but how do we get it in? Well, the air that we breathe in has to come in and down through our trachea, and our trachea are obviously lined with our ciliated epithelial tissue. Now, ciliated epithelial tissue could be asked in paper 1, it's the transport exchange system, or it could be asked in the paper 2 with regards to its role in the immune system. So it really does depend. It could be module 2, how has the cell differentiated? So it could come up in any of the papers. So our ciliated epithelial cells are going to beat in rhythm, and they're going to beat to move the mucus, which is obviously there to trap the pathogen, stopping it from getting into the lungs. The way in which it beats is to do with the cytoskeleton. The fact that the cytoskeleton is able to allow for the movement of the cell, that is what gives that rhythmic beating of the cilia. Obviously, our goblet cell is the one that's going to secrete the mucus, and mucus is made up of water, carbohydrates, proteins, triglycerides. So if they're asking about the mucus, are they asking about the production of the proteins? If so, ribosomes. Are they asking about the production of carbohydrates? If so, smooth endoplasmic reticulum. So make sure, depending on if they are specific, which one they're looking at in the question. And we obviously know about our cartilage rings. Our cartilage rings are made up of collagen and elastin fibers. So if they're made up of collagen and elastin fibers, we know these are fibrous proteins. Fibrous proteins which are flexible, which are strong, preventing the collapsing of the trachea. So allowing that exchange and the ventilation. So if we now come down, we've got through our trachea, we obviously go to our bronchi, which is plural, bronchus if it's singular. And then we have our bronchioles, the little branches at the end. And we need to remember, again, depends if it's going to be in paper 2, if it's going to be in paper 1. If we have a way in which we are going to expulsive reflex. So the expulsive reflex is a reflex which is animal response. There is some form of stimulus that is detected that is then going to trigger a response which is a unconscious response. It is autonomic, and so therefore it's a series of these coordinated actions to remove anything which is unwanted or harmful inside the lungs. So if I breathe in dust, I am then going to have my smooth muscles in the bronchioles constricting and restricting the airways. So the bronchioles constrict, that therefore stops as much of the airflow going in and therefore protects my lungs. If I need to cough or sneeze, if I've ingested something, these are all examples of the expulsive reflex. And again, it depends where they're going to, if they're going to ask a question on it, which paper, because it could be in the exchange system or it could be in immune system. Obviously, we get to the end of the mammalian ventilation system. We're looking at our alveoli, that site of gas exchange. Now, our ribs and our intercostal muscles are important here in the fact that we need to know whether we're talking about our external intercostal muscles or whether we're talking about our internal intercostal muscles. So if we are talking about our external intercostal muscles and they are contracting, they're going to lift our rib cage up and out. That is therefore going to change the volume. It's going to increase the volume. If we are going to contract our diaphragm, that's going to drop it down. That is also going to increase the volume. What would happen to the mechanism of smooth muscle if vomiting occurs? Well, the vomiting is going to occur where we're going to squeeze, therefore constrict the muscles that's forcing at a high velocity out, isn't it? So the vomiting would be a signal that is being sent to the effector being the muscle. The muscle is responding by contracting, and that contraction is going to force the fluid, which is inside, and the substances inside your stomach, up through your esophagus so that you can then get it out of the body. So it's a protective mechanism that's going to happen. Okay, so if we're looking at our alveoli, this site of gas exchange, we know we've got our epithelial layer, we know we've got our endothelial layer, we know that these are very squash cells. These cells have a short diffusion distance. So our squamous epithelial cells are in our alveoli, and we have our endothelial cells in our capillary, and therefore, because each of them are one cell thick, we have a short diffusion distance. The gases are going to diffuse from a high concentration to a lower concentration. Now, we also have the fact that our alveoli are going to have this surfactant which is put there. Now, we know that water has a surface tension, and because water has a surface tension, we need to actually prevent our alveoli from collapsing. If our alveoli collapse, there is not as much surface area, therefore there won't be as much exchange. So for surfactants are these layers of phospholipids which are put onto the surface of the water, and they stop that collapsing of the alveoli so that we can increase the surface area. Obviously, we have ventilation. We're going to remove the stale air. We're going to bring in fresh air, therefore maintaining a concentration gradient so that that exchange can happen. And also, we're going to ensure that there is a good blood supply by having the blood capillaries running alongside all the alveoli. So if we were looking at our gaseous exchange, we know as you sit here right now listening to me, your lungs are doing this. They are expanding and contracting, and that is all to do with our external intercostal muscles contracting, our diaphragm contracting, therefore our volume inside our thoracic cavity is going to increase. Because that increases, the pressure is going to decrease, and that pressure is going to decrease lower than the atmospheric pressure, and therefore air is going to flow into our lungs. If we are exhalating, if we are breathing out, that is where our external intercostal muscles are going to relax. So as our external intercostal muscles are relaxing, that also will have our diaphragm relaxing, and that will result in the elastin fibers. Now remember their ability to stretch and recoil. The elastin fibers are going to recoil back to their original position, and as they do that, it's going to therefore decrease the volume, increase the pressure, and force the air out. What does it look like if the diaphragm contracts? The diaphragm contracts, drops down. So what it will do is it will drop down, it will be more flattened than it would be. So if you look at this one here, it's very dome-like shaped, whereas this one is flattened down. So when it contracts, it flattens, therefore increasing the volume. When it is contracting, when it's contracting, it drops down. When it's relaxing, it gets back up into that dome-like shape. So you should know about how to read a spirograph, a trace reading even from a spirometer. So you should know your tidal volume, which is this up and down, which is your normal breathing. If I take a deep breath in, that's going to go up, and then I can go back to a normal breathing. If I really try and force the air out, that is my expiratory reserve volume. And if I I'm looking at both my inspiratory and my expiratory. I am therefore joining them together. That is my vital capacity. So you should know how to read your total lung capacity and reading it off the graph, looking at, for example, the readings. Oh, this is where I don't have the one thing. Oh, yes, I do. Hold on. I do. So, Khadija, you've asked about what do I mean by elastic fibers recoiling. So, my hair bubble here is made up of elastic fibers, right? So, we've got inside elastic, which has a material on the top to protect my hair. So, if you look at elastic bands, they're made up of elastic. So, if I stretch, if I open it, I'm stretching it, what's happening? So, if I now do it from the outside, if I stretch it and I let go, what's going to happen is it's going to recoil back into its original position. Now, as I recoil it, it's going to potentially, it may end up coming back and twisting, but that's where you've got the surfactant. So it can stretch and recoil back to that original position, and therefore my lungs, as my rib cage is being lifted up and out, that is going to stretch. As the muscles are relaxing and the elastin fibers are recoiling, that's going to bring my rib cage back in and down. So the lifting up and out of the rib cage is my external intercostal muscles lifting up and out. My elastin fibers are recoiling, which pulls my lungs back down to the original position. And so therefore, if I am breathing normally, this is happening at a resting rate, which is my breathing rate. So you would look at how many peaks there are or how many troughs there are in a given time. So make sure when you are reading the trace, again, that you are looking at if they've given you a time or if they are giving you the lung volume, what's the volume that's been measured? Oh, hi guys. Hope chemistry went well. I've just seen someone else say that the chemistry paper 1 went well. Now, for chemistry, OCR chemistry, that actually normally means that one of the other two papers is going to be harder. Sorry to give you that. There's always going to be one of the papers which is much harder and one paper that's much easier. So if the first paper's gone well, watch out for those harder questions that are coming later on. But it's good to come out feeling that it was challenging, but okay. I have heard about the accessibility of the exam and the fact that they're going to, for example, have your numbers, temperature going from 0, chronological order, things like that. Yes, that has been put out that they're going to ensure that the papers are made in a easier format than what they have done in the past. Yes, I had heard that. Okay, so switching over then, we're going to go from the ventilation system and we're now going to look at the circulatory system, and then I will link them back in again to do with the animal response. So we should know that we have a circulatory system, and our circulatory system is a closed double circulatory system, and that is so that it can ensure that the blood that is being pumped around, because we are multicellular, because we are larger, more complex organisms, we have a higher metabolic demand. We're also endotherms. We have a higher metabolic demand. And so therefore we need a closed system. We need a transport system that is going to deliver the oxygen and remove the carbon dioxide in the right efficient way. So we know at the alveoli what's going to happen is our oxygen will diffuse. Our oxygen will then be able to bind to our hemoglobin, and when it binds the hemoglobin, what will then happen is the Oxygen binding can then release the hydrogen ions. Hydrogen ions will then be able to bind with hydrogen carbonate ions to form carbonic acid. That will then be able to be converted into carbon dioxide and water. The carbon dioxide can then diffuse from where there's a high concentration to where there's a low concentration. And when we look down at the respiring tissue, we know that carbon dioxide is being produced because of decarboxylation. We know that the oxygen is being used as a last electron acceptor. So the partial pressure of oxygen at respiring tissue is normally low, and therefore, because the partial pressure of oxygen is low, what's going to happen is the oxygen is going to be dissociated, and that will therefore release the oxygen. The oxygen can then diffuse from a high concentration to a lower concentration and be used inside the mitochondria at the cristae for oxidative phosphorylation. When we're up at the lungs, the partial pressure of oxygen is always higher than the partial pressure of oxygen inside the blood, and therefore that allows for the diffusion. So you should know both ways in which this could go. I haven't covered respiration, it's still coming, so don't worry, that is still coming, but it's where could they link it together. So whenever I am doing these topic areas, whenever I'm talking about respiring cells or respiring tissue, I would always be going, oh, the oxygen is needed for aerobic respiration as the last electron acceptor in oxidative phosphorylation. I'm therefore giving the detail of what is important. Now, I do that in the stem of the question. When I am writing little notes to myself as I'm looking at the question, I would be writing these little notes. I would then ensure when I formulate my answer that I don't give information that's irrelevant to the question. That is going to limit your marking points if it's a 6 marker. If I give irrelevant information and I just blurt it on paper, I'm not going to get the marks. If I have done it as a way of priming myself, priming my brain, that's okay if I'm doing it in the stem of the question. But in my answer that goes in the answer line, I have taken the time to look at what I've written and which bits are relevant to the question that's being asked. That's what's important. That means that is going to help you in how to get and gain those extra marks. So important ways of looking at this, whenever you look at a dissociation curve, and I do feel like one is coming, if we're looking at a dissociation curve, if I am looking at my partial pressure of oxygen, when I am at normal cells, my partial pressure of oxygen is between 5 to 7 kilopascals. When I'm very active, when my cells are doing lots of aerobic respiration, that partial pressure is going to be 3 to 5 kilopascals. So when I read my dissociation curve, what I'm looking at is I'm looking at, okay, if I were 25% saturated, what would that partial pressure be? If I were 50% saturated, what would that partial pressure be? 75% saturated, what would that partial pressure be? Now, obviously, in the exam, you will use a ruler. Do not freehand like I've just done. I'm doing it on my screen. That makes it slightly different. So I would say you must have a ruler. You must have a sharp pencil. You must be using the graph to look at what they've told you if they're asking a question on it and try to use that in your answer. So if I am going to have a very active cell, I have lots of respiration happening, I have high carbon dioxide. So where is the carbon dioxide being produced? It's being produced in the Krebs cycle and it's being produced in the link reaction. So I know that through the process of decarboxylation, which happens both in the Krebs and the Link reaction, that will therefore produce lots of carbon dioxide. Carbon dioxide is then going to diffuse, and if there is high partial pressure of carbon dioxide at the respiring tissue, that is going to therefore lower the pH, because we just said CO2 plus H2O is going to react together to form our carbonic acid. The carbonic acid will then dissociate into our hydrogen ions. Oh, let's do that again. And our hydrogen carbonate ions. And because it's going to dissociate, that will therefore promote the unloading of oxygen. So if the partial pressure, let's say this one is 3.5 kilopascals, it was 25% saturated. Now it's about half. It's about 12.5, but obviously in the exam you would read whatever the scale is. So at the same partial pressure, the hemoglobin is less saturated. And I can do the same if I go to 50, on normal, and then I look at my high partial pressure of carbon dioxide. That is therefore going to dissociate more readily. So it's now only, maybe 30% saturated. So what we can see is whenever we are looking at carbon dioxide, we have our dissociation curve, same shape, but it has been shifted to the right, and we call that the Bohr shift. And then if I go for the opposite side of things, if I'm looking at a increase in the affinity for oxygen, then I would do exactly the same thing. At 50% saturation would be at my 3.5, whereas that one is less than 25% saturated. So when we talk about fetal hemoglobin, because the partial pressure of oxygen is always low in the placenta, that basically means that the oxygen will dissociate. It will be released from the adult hemoglobin. It will then diffuse across, and when it diffuses across the placenta, it will then be loaded onto the fetal hemoglobin. What does the y-axis mean? The percentage saturation of hemoglobin. So basically, if I have, let me think this one through. So if I had 10 hemoglobin molecules, okay? So if I have 10 hemoglobin molecules, if I can spell, might help. And I know I'm really bad at spelling, so just be aware of that. If you are like me and you are very unlucky because your brain works too fast and you write things slightly the wrong way around, then I would say try and slow yourself down during the exam. Try and double check your answers. So if I have 10 hemoglobin molecules and I know that each hemoglobin molecule can carry 4 oxygens, so 4 oxygen molecules can be carried by 1 hemoglobin molecule. If I am 100% saturated with oxygen, that means all 10 hemoglobin molecules have got 4 oxygen molecules. Does that make sense? And then if I have the 10 hemoglobin molecules, but this time instead of having 4 oxygen molecules, they only have 2 oxygen molecules, that would therefore mean that they were 50% saturated. I hope that kind of makes it make sense. But when you're looking at a graph, you want to be describing at that partial pressure what is the percentage saturation using the terms. Okay, so now we're going to look at coming back to animal response and homeostasis. So if I am looking at an increase in my carbon dioxide concentration, because the increase in my carbon dioxide concentration has occurred due to an increase in exercise, an increase in metabolic reaction, whatever it is, we have got chemoreceptors that are going to detect the changes in the blood, and they're going to detect the carbon dioxide concentration, they're going to detect the pH. So they're looking at two things. What's the carbon dioxide concentration? What is the hydrogen ion concentration? So we know that our tissue will be doing the respiration. We know that it is going to diffuse into the blood, and we know that it is going to be transported. So if we have an increase in our carbon dioxide concentration and we have a decrease in our pH, we know that we need to remove that carbon dioxide, because if it becomes too acidic, that's going to have a big knock-on effect to our proteins, our globular proteins that are sensitive to pH. So therefore, when it has been detected by the receptors, that will then result in the coordination center then passing that information on to the effector then carrying out the response. So if we are having an increased carbon dioxide and decrease in the pH, we're going to increase our breathing rate. We're going to increase the rate at which the diaphragm and external intercostal muscles contract, and that will mean that our breathing rate will increase and the depth of the breath will also change. And also what we can do is dilate our bronchioles. And if we dilate our bronchioles, it means it's like opening a window a small amount or opening a window a large amount. If I'm dilating, I can have more air flowing in. Go the opposite way, because remember, there's two sides of this. If I need it to go the opposite way, if I have a decrease in carbon dioxide, if I have an increase in the pH, my breathing rate needs to decrease, because the contraction of the muscles is going to obviously need energy. That energy is therefore going to be produced. I just said produced, and I should never say that. That energy is going to be released after we have hydrolyzed the ATP down, and therefore that ATP is needing to be produced through the process of respiration. So if I don't need to have this contraction happening at a faster rate, I need to slow it down. And so therefore my breathing rate at rest is actually signaled because of my parasympathetic nervous system. So if it is my sympathetic nervous system, remember we talked about the fact that that would be noradrenaline that is going to bind. That's a neurotransmitter that's going to be released. If it's my parasympathetic, it's acetylcholine. So this is where we kind of done a circle back depending on where they want to go. Obviously, we're using negative feedback. Now, with my heart, if I'm going to increase the circulation of the blood, I need my heart to pump faster. Now I'm going to go through this one quite quickly because I don't feel like heart's coming up that much. I could be wrong, but I don't want to skip it out. But we know cardiac muscle is striated, we know that it's branched, we know that it's uninucleated, and we know that it will synchronize its contractions together. So because of the SEN, the contractions of the atria are together as one. That is your atrial systole. Your AVN is then going to pass the depolarization onto the bundle of His, down the Purkinje fibers, and we're going to contract from the ventricles at the apex up. We're going to push and force the blood out. So we know that the SEN is this highly specialized cell. We know that that's going to depolarize on its own spontaneously, but we can speed it up and we can slow it down depending on what is required by this movement of the molecules because of the rate at which we are respiring. So if we are going to increase our heart rate, remember, and I do think if they're going to ask anything, they could ask you to do a calculation on this. Your cardiac output is calculated by your heart rate times your stroke volume. So your cardiac output is your heart rate times your stroke volume, and therefore if I increase my heart rate, then I could actually decrease the stroke volume that's coming out and still have the same cardiac output. So they'll give you potentially either the heart rate or the stroke volume and the cardiac output and ask you to rearrange an equation there. If we know about our receptors, linking back to the animal response, we have receptors that are going to be detecting. These are mechanoreceptors if we're looking at blood pressure, if we're looking at carbon dioxide concentration, they're chemoreceptors. They will therefore have a way of detecting whatever it is they're detecting. They will send the impulse to the medulla oblongata. The medulla oblongata will then either send it to the sympathetic nerve, to the SAN, which would then increase the frequency of the heartbeat, increase the frequency of the impulse from the SAN, shorten the delay between the SAN and AVN, increase the force of contraction, increase the blood supply to the heart. If we go in the opposite way, if we go in the parasympathetic nervous system, we'll do the opposite. We'll decrease it, decrease the frequency, lengthen the delay. This is part of your autonomic nervous system, and therefore, depending on which one is going to release the neurotransmitter, depends on how that is responded to. So if they don't do that one, they could look at the pancreas. And I, again, do feel like homeostasis is coming through. So if they give you the pancreas, can you identify the islets of Langerhans, which are always lighter? If you're looking at islets of Langerhans, which are lighter, you therefore know that they are using differential staining. So they're going to differentially stain so they can identify in this histology. Here is the islets of Langerhans. Here are the acinar cells. The acinar cells are more active in the production of enzymes because they make your digestive enzymes, and therefore we are seeing them be a darker color because they've got more of the rough endoplasmic reticulum. We can make that assumption because it's a dark purple color. So one of the things that they could do is ask you to interpret a picture. So what actually happens at the islets of Langerhans, we're going to talk about the insulin secretion that occurs at the beta cells. So if I have low glucose concentration, I know I've got my sodium potassium pump, I know I've got my resting state of my cell, and so therefore if I have low glucose concentration, my potassium is going to be able to leave through a potassium channel, which is going to maintain my membrane at a hyperpolarized state, and that hyperpolarized state therefore means that my voltage gated calcium channels are going to be closed. There is going to be no secretion of my insulin because I am nowhere near my threshold value. So that is one way in which they could assess that understanding of potential difference across the membrane, and understanding how these voltage gated calcium channels, if they're not open, if they're closed, they're not going to allow the calcium to move. Now, if the calcium is moving, which way is it moving? And that's important of if they want to assess your ability to talk about biological membranes. So high glucose concentration, we have the obviously high potassium levels that are happening. However, glucose is broken down by glycolysis in the cytoplasm. Then we've got the Lynx Krebs oxidative phosphorylation. We've got aerobic respiration happening. We therefore have ATP production. ATP will bind to the channel here. When it binds to the potassium ATP channel, it will close it. Because it's closing it, the potassium can no longer leave. That is going to depolarize the membrane. If it reaches the threshold value, we know these voltage gated calcium channels can open. We know the calcium moves in, so we can definitely say that there is a high concentration outside in comparison to inside. When the calcium comes in, that will then result in the insulin being able to be secreted by exocytosis. Yes, it does have chi-squared in there. Try the patterns of inheritance videos. So if insulin is secreted from the pancreas, key thing for you to know is to not forget the connectivity of the tropic areas. So the pancreas is going to secrete the insulin. The insulin is then going to be transported from the place where it is secreted to where it's going to have its target. And the target is going to be on the liver cells, for example. So, or it could be the muscle cells, depending on where the question goes. You should know about your liver. You should know about hepatic vein. You should know about the hepatic portal vein. They are veins, therefore they will have large lumens. They will have thinner layers, but they'll still have the 3 layers. The hepatic artery is going to carry your oxygenated blood to the liver. The hepatic vein is going to return the deoxygenated blood from the liver to the heart. The hepatic portal vein is the connection between the intestines, the pancreas, and the spleen to the liver, so that the liver can filter out any of the things that are required. So if we look inside the liver, then we know we've got our sinusoid, we know we've got our hepatic portal vein, we know we've got our hepatic artery, we know we've got our hepatocytes, and we know we've got our hepatic central vein. So we should know that when the blood is moving through, substances are going to be exchanged where there is that capillary. So the exchange that is happening is where they are moving out of the blood into the hepatocytes, and the hepatocytes are going to be doing with them whatever needs to be done. So we know our hepatocytes are multipotent, therefore they can divide and go back into they can repair themselves. So in the liver, because they are multipotent, they have the ability to divide and differentiate. They can repair any damage that may have done. If it's more saturated, does that mean the oxygen dissociates more readily? When you talk about if it's more saturated, is the saturation, I'm trying to work out what you mean. Is it more saturated with carbon dioxide, more saturated with oxygen? What you're looking at is if the hemoglobin is saturated, then it's holding on to, it's loaded it and holding on to the oxygen. If it is if the saturation is lower, the percent saturation is lower, it has dissociated, it has unloaded the oxygen. So it depends what you mean by if it's more saturated. Okay, one of the things to know about your hepatocytes, again, if they ask you, does the cell have a large nucleus? Does the cell have prominent Golgi apparatus? Does the cell have lots of mitochondria? If it has large nuclei, that means that it has got lots of protein synthesis occurring, and therefore it is going to be doing lots of transcription, translation that is required by the cell, because obviously this is a cell that does a lot of filtering, it does a lot of detoxification. It's going to require to have the right amount of ATP. It's going to require the Golgi apparatus for the modifying and packaging of the substances. The Kupffer cell is a type of macrophage which is going to engulf and digest any of the debris of cells, but also these ones stay and patrol in the liver so that they can get rid of any erythrocytes which have been degraded. And then we have the bile duct. So the hepatocytes are used to produce bile, and bile goes into the bile duct to go to the intestines to help with the emulsification of the lipids. Okay, I'm very aware of the time, so I'm going to try and really speed through this. If they talk about glycogen storage, do you know about insulin? Do you know that the insulin, obviously released by that pancreatic beta cell, is going to activate expression of glucose transporters, and that will therefore increase the uptake of the glucose, and the glucose will then be converted into the glycogen through the process of a condensation reaction. So remember, your glycogen is a biological molecule. You have that it's branched, is insoluble, it's metabolically inactive. Try and give these key things if they're asking for a 6 marker on glycogen. If we're talking about respiration, we know that through the process of glycolysis, the glucose is broken down into pyruvate. We know the pyruvate will then enter into the mitochondria if oxygen is present, and we get our link reaction. We then have our Krebs cycle, and then we have our oxidative phosphorylation. So what's the point in this enzyme controlled reaction? The process of this enzyme controlled reaction is to produce lots and lots, or to not produce, but donate lots and lots of hydrogens that can be taken to the electron transport chain for oxidative phosphorylation. And so if oxygen is present, pyruvate enters the mitochondrion. If the oxygen is not present, then the pyruvate will go through anaerobic respiration, and depending on whether we're talking about yeast or mammals, depends on whether we're doing alcoholic fermentation or whether we're doing lactate fermentation. I do feel like anaerobic respiration is coming up, but I'm not sure whether it's going to be in paper 1 or paper 2 linked to biotechnology, or whether it's going to be in paper 3, but I do feel like that's coming through. So you should know the stages of glycolysis. You should know about the phosphorylation. You should know about the lysis. You should know about the dehydrogenation. You should know about the substrate level phosphorylation. You should know about link reaction and decarboxylation and oxidation. You should know about the Krebs cycle, and you should know the names of your not what I meant to do. Your oxaloacetate, your citrate, your acetyl group. I'm not sure they're going to ask that much on it this year because they've done so much the last couple of years, but it doesn't mean you shouldn't know it. Please do be aware of all the different parts of it, where they occur, what are the products that you're going to get. Now, if we've got aerobic respiration, we know we've got these 4 processes happening. Now, glucose is the respiratory substrate of choice. However, if glucose is not available, or if lipids are highly available, then the organism can utilize the lipids. So remember, our lipids are glycerol, 1 glycerol, 3 fatty acid chains if we're looking at a triglyceride. If we then utilize that, we can convert glycerol into pyruvate. We can break the fatty acid chains down into acetyl groups if we have the right enzymes, and by doing that, that therefore means we can then continue doing, for example, the Krebs cycle and oxidative phosphorylation, or links and Krebs and oxidative phosphorylation, and that is going to therefore produce less carbon dioxide for the amount of oxygen that is going to need to be consumed. So your respiratory quotient, and they did ask a big question on this last year, you should know your numbers 1, 0.7, 0.9. You should know that with amino acids, we are going to have to deaminate them. That happens at the liver, and the remaining carbon skeleton can then enter, depending on what the R group was, depends on where it will actually enter. So again, they have asked a question about this in the past, where it enters at different points, and it asks you about how many times it can, or how easy it would be to convert it to a molecule within the Krebs cycle. If we use lipids, it skips glycolysis and link reaction. Yes, but you do need the enzymes that are going to be used to break down to produce the acetyl groups. So you would be breaking down the fatty acid chain into 2 carbon acetyl groups. So if you had a chain which had, for example, 50 carbons, then you could produce a high number of those acetyl groups. So if they are going to ask you a question, they may expect you to be able to work out your carbon dioxide produced over your oxygen consumed, and therefore figure out which of the respiratory substrates are being used by whatever it is you're using. It could be germinating peas, it could be woodlice, it could be sunflower seeds, whatever that may be. They could ask you about a respirometer, and I do have a strong feeling that you're either going to be asked about a respirometer, or you're going to be asked about planning an investigation to do with a respirometer. So make sure you understand that with the respirometer, you've got oxygen going in, carbon dioxide being absorbed by the soda lime. We would expect the distance to be moved If we know our capillary tube diameter, we can therefore work out our volume, and they may ask you to do a maths question on that. Obviously, if we're going to use wood lice, we need to prevent organisms touching the soil line. We want to not harm them. If we are using a manometer, we would want to use glass beads. If we were just using a simple respirometer, we would do it with a respiring organism, and then also do it with either glass beads or beads that have been boiled. One second. So we would want to do it with beads that have been boiled, and the reason we're using beads that have been boiled is because we want to make sure that they are not able to carry out the respiration, because we want to see what happens if it wasn't. If the RQ is 0.8, what would the substrate be? If it's 0.8, you are therefore saying it is a mix of carbohydrates and lipids. The closer it is to 1, mainly carbohydrates. The closer it is to 0.7, mainly lipids. If it's somewhere in between, if it's animals, then we are saying that it's a mix of the two. If it is plants, we could, and even they did a question on ants before, you could get the marking point for saying that it's proteins, but it's dependent on the organism, because proteins, if we start to utilize proteins, we are therefore going to be potentially damaging tissue. So it's about knowing where it lies, and that it would be a combination of the two. So yeah, if we're doing this, we would obviously expect that to move, and we'd want to calibrate it, and this would be used as a control. Now, they could link that to your reducing sugar test. So obviously, you know you've got your Benedicts, you know you've got your non-reducing sugar test. You should know the key things. You should know how to do a serial dilution. So you should know if we're going to use a Benedict's reagent, or if we're going to use a biuret reagent for protein, or if we're going to look at a ethanol emulsion test, then we're going to need to make sure we have known concentrations. If we have known concentrations, we're then going to want to ensure that we've got a calibration curve. So make sure you know how to do your dilutions. If we're going to use a colorimeter, we're looking at ensuring that we understand that the light source is going to then be stopped by a filter. We're therefore going to use a filter which is complementary to whatever the solution is that we're using. So if it's Benedict's, we use a red filter. If it was biuret, we'd use a green, a yellow or a green. If it is emulsion, we'd use a blue. If it is beetroot, we'd use a green. If you are unsure, write a complementary color. They will give you that unless it's Benedict's. They do expect you to know the red filter, but they're more lenient with the other ones. Obviously, you need to zero so that you can reduce the systematic error and know whether you're using the absorbance reading or whether you are reading the transmission. So if they're going to give you a calibration curve, make sure you know how to describe and how to explain, okay? And if we go back then to our glucose and we talk about our insulin and our other hormone, which is glucagon, we know that we have got homeostasis. We need to keep our blood sugar levels at the right level because of the respiration. We need glucose as a respiratory substrate of choice. Also, we need to maintain our water potential. So we want to maintain our blood sugar levels at a specific narrow range, and so therefore we have insulin when our blood sugar levels are high. We have glucose agon from our alpha cells when our blood sugar levels are too low. Obviously, they are hormones which are secreted from glands. They are then going to be transported in the bloodstream to the target cells that they are going to work on. And what's going to happen if it's insulin? We're going to increase glucose absorption. We're going to increase respiration. We're going to have glycogenesis, so therefore we're producing glycogen. We're going to have lipogenesis. We're going to produce lipids. If we are having low blood sugar levels, we are therefore going to have glucagon released when that binds to the liver. That is therefore going to cause glycogenolysis. We're going to hydrolyze the glycogen down. We're going to reduce the glucose absorption. And we are also going to potentially produce glucose from other substances such as the glycerol or the proteins, the carbon skeletons. So what is this an example of? Negative feedback. It's negative feedback. So if they give you a graph and the graph is going up and down and up and down and up and down in your exam, they're expecting you to be able to know when it's going up, you have absorbed the glucose. That glucose needs to then be responded to, and that will therefore result in the release of insulin. If your blood sugar levels are going down because the insulin is being responded to, when it gets too low, just before it's about to come back up again, that's when you've got your alpha cells releasing their glucagon. So up and down, I can see a graph coming where you're going to have to interpret that. Obviously, there's two different types of diabetes. If it's type 1 or type 2, make sure you look at what they've told you. If it's type 1, it's to do with insulin being injected. If it's type 2, it's about the insulin being not responded to. So the beta cells are not destroyed. They're still making the insulin, but they are not being responded to. 1 second. Turn that off. Okay, so obviously, the liver is also going to do detoxification. If it's detoxifying, for example, alcohol, we need the enzymes to convert the ethanol back into the less toxic substance, fatty acids, and cellular respiration. So if we are looking at the production of alcohol, that I feel like it could come into paper 2 in biotechnology. Because it's anaerobic respiration, we know that it's pyruvate to ethanol, ethanol to ethanol, so that the glycolysis can keep happening. It's anaerobic because it's not in the presence of the oxygen. Okay. Wow. I can do this. I can keep going. Okay. So, formation of urea then. Deamination, we're going to remove it. We're going to make ammonia. Ammonia is toxic. We're going to use the ornithine cycle to produce urea. Urea is toxic if it's in a high quantity. So therefore, we need to get rid of it. And as I just said, the carbon skeleton can then be used in other ways. If they're going to ask about the formation of urea, it will be about you saying that ammonia is converted into urea, and that is done through an enzyme controlled reactions known as the ornithine cycle. When they gave a question on this before, what they then did was they were looking at transport across this organelle, which was the mitochondrion. And so therefore we're crossing two membranes. If we're crossing two membranes, we have to know about the transport of molecules. So don't get panicked about a diagram like this going, I wasn't told about the ornithine cycle. You're not meant to. It's application. If I've got adenosine monophosphate being produced here, then that has to be ATP. So there is an expectation that you are able to apply your knowledge, and obviously urea is then excreted by the kidneys. So you should know the kidneys. You should know glomerulus is a capillary, and that is then going to be at a high pressure. I'm still going strong. I am. I'm on my just over 3.5 hours now. So the high hydrostatic pressure in the glomerulus is going to force the substances out into the Bowman's capsule. So make sure that you are able to identify where the Bowman's capsule is. Make sure you know that the glomerulus, Bowman's capsule, proximal convoluted tubule, distal convoluted tubule, all of these are found within the cortex. Somebody asked about oncotic pressure and hydrostatic pressure. So the key thing to note, whether we're talking about No, it wasn't adenosine diphosphate. It was adenosine triphosphate they wanted you to say, because it was used in that reaction. So they were specific. They wanted adenosine triphosphate, not diphosphate. You would assume that it would go from adenosine triphosphate to di to mono, but when we look at cyclic AMP, we are therefore using a specific enzyme which does that conversion. So no, it was expecting ATP in that question. When we look at tissue fluid formation or ultrafiltration, we need to understand hydrostatic pressure. So if I have hydrostatic pressure, which is 4.3 kilopascals pushing out, and I have hydrostatic pressure from my tissue fluid pushing back in, the overall difference is there is a net of, in this case, 3.3 kilopascals pushing out. So the net overall force is going to push the fluid out of the capillary into the tissue fluid. When we look at the oncotic pressure, we're talking about the fact that there are plasma proteins that are going to maintain the water potential inside the blood capillary. Remember, it's a closed circulatory system. So we are maintaining the blood capillary oncotic pressure at a minus 3.3 kilopascals. The tissue fluid has its own oncotic pressure, and that is going to obviously be a net in which we have minus 2.1 kilopascals. So we have hydrostatic pressure pushing out, and we have oncotic pressure pulling back in. So when the hydrostatic pressure pushing out is greater than the oncotic pressure pulling back in, that is therefore going to force more fluids out of the capillary. When you get to the venule end, the difference between the hydrostatic pressure and the oncotic pressure results in there being a lower hydrostatic pressure. I'm just trying to find, there we go, this slide. So if we're at the venule end, we would therefore have a net hydrostatic pressure, which is, for example, 0.5 kilopascals. We still have minus 2.2 or 2.1 kilopascals pulling back in. So we're pushing out with 0.5 kilopascals, but we're pulling back in with minus 2.1 kilopascals. So overall, the oncotic pressure is greater than the hydrostatic pressure. So the fluids are being moved back into the capillary. So if you understand that, then hopefully that will help you when it comes to the next part, hold on one second, where we're looking at the glomerulus. So high hydrostatic pressure, we have a basement membrane preventing the substances from going across, and then we have our podocytes, which are differentiated cells, and those differentiated cells are going to act as another filtering layer, preventing the substances from moving in. If we're looking at our proximal convoluted tubule or our distal convoluted tubule, they are very similar. So site of selective reabsorption, the proximal has microvilli, which would increase the surface area. We know we've got active transport of our sodium potassium pump. We know we've got co-transportation of sodium, ions and glucose. If we're looking at the distal convoluted tubule, we would therefore be saying again selective reabsorption, but this time of mineral ions. We would be talking about no microvilli. We would be saying active transport, co-transport protein, and therefore because it's co-transport, this time it's sodium and calcium. They can decide what they're going to give you as the example for the distal convoluted tubule. I don't know why, but I feel that they may bring in aldosterone and they may bring in the control of water with the hormonal side of things. So if we have too much water or too little salts, our blood pressure is going to rise, higher volume of blood detected in the carotid artery. So we've got our mechanoreceptors. Therefore, we've got our coordination center, which is going to result in our adrenal gland, which is going to secrete adrenaline from No, let's try that again. It's going to secrete aldosterone from the adrenal cortex. Aldosterone is going to result in more sodium channels being inserted into the distal convoluted tubule, and therefore more sodium ions can be reabsorbed. We have the sodium concentration in the blood tends to rise, and therefore we get the correct water potential. Go the opposite way, we do the opposite. We're going to have less secretion, we're going to have less sodium channels, therefore we're going to have less sodium in the blood. And so therefore, again, negative feedback, but looking at it in our salt concentration. You should know about your mineral corticoids, and aldosterone is a prime example of that. When we look at the lip of Henle, they could go on the lip of Henle and talk about the fact that our ascending limb is the place where we're going to actively pump our sodium chlorides out. We've got our descending limb where we're going to have our water being able to move because of that permeability. And therefore, if we are looking at osmoregulation, we need to then bring in our collecting duct as well. So if, just going back up here, if we're looking at the, she says, hold on, I want to bring, go back up a second here. If we're looking at our lip of Henle and our collecting duct, if our medulla is very thick, we can assume that there is a longer lip of Henle. If there's a longer lip of Henle, therefore we would have more areas where we can pump the salt out, therefore more areas where we can move the water out of. Yes, we would. I'm just about to come to that now. So if we are looking at our antidiuretic hormone, which is obviously produced in the hypothalamus, and it is going to be secreted from the posterior pituitary gland, we therefore need to look at our osmoreceptors. So osmoreceptors in the hypothalamus are going to monitor our inorganic ion concentration in the blood, and that is going to then result in the posterior pituitary gland secreting more or less ADH depending on what needs to happen. So if we have got our water potential being low, water will leave by osmosis, resulting in a signal being sent to that posterior to release the ADH. The ADH is then transported in the bloodstream to the collecting duct. We also have our baroreceptors in the aortic and carotid artery monitoring our blood pressure. If we have a high pressure, we therefore have too much water, that would also signal to the kidneys and what needs to happen. So ADH gets to the collecting duct, it will bind, it will then activate adenylate cyclase. We then have our secondary messenger, which activates our protein kinases, which therefore causes our vesicles to fuse with the membrane and therefore makes it more permeable. So when we look at osmoregulation, when we look at the kidneys, we are talking about unfiltered blood being filtered, but we are going to selectively reabsorb at different points, and we are therefore going to produce urine, which we need to excrete with our nitrogenous waste, and we are going to regulate the blood which is leaving through the renal vein. And obviously that urea, which was produced through the ornithine cycle in the liver, is going to be excreted in our urine, and the concentration of our urine is dependent on how much water was being reabsorbed back in. So if you drink lots of water and you produce large volumes of urine, that is going to be because you are not needing to reabsorb it back in, because we need to maintain that water potential. And then the other thing to note about animal response is if there's a threat detected by your autonomic nervous system, that will result in communications with your parasympathetic system or your sympathetic system. Which one? What are we going to do? Well, the noradrenaline will obviously go with the muscles and the glands, but we also have that our noradrenaline can signal for the medulla of the adrenal gland to secrete adrenaline or noradrenaline. And so these hormones are going to bind to a receptor which is specific for it at its target cell. So be aware they could go down the neuronal communication or they could go down the hormonal communication. Make sure you know what happens with the adrenaline, about that activation, about the cyclic AMP, which is our secondary messenger, and about the fact our protein kinases, in this case, is going to convert our glycogen into glucose. So we're going to break this down because it's branched. It's got lots of places where it can be broken down. Other thing to know, if they are going to go hormonal system, to know whether they are asking about it being a peptide. If it's a peptide, it cannot cross the phospholipid bilayer. If it is a steroid hormone or a non-polar molecule, it can get across, and that could result in the activation of gene expression, which then could activate enzymes. It really depends where they want to go with the question. Okay, right, I'm going to have to really speed through the plant stuff. I don't know why I have left this to the end, but here we go. I can do this. I'll go a little bit longer than I planned to. I was going to stop at 2, but I'm going to keep going until, or as long as I can. I'm going to see if I can finish these slides. Okay. Obviously, we have got our eukaryotic cells, we've got our plant cells. We know with our plant cells, we have got some extra parts to think about. Our chloroplast, which is our site of photosynthesis. We have a permanent vacuole, and that's going to contain cell sap, which is important for maintaining turgor. Remember, plants are able to take in water. That water means the cell can push against the cell wall. The cell wall is made up of cellulose, a polysaccharide. Because it's made up of this polysaccharide, it therefore means that it maintains the shape, the rigidity. And so by the permanent vacuole being able to contain cell sap, having a water potential, it is therefore going to maintain that turgor pressure by maintaining the water inside. So what you have to remember is the permanent vacuole has a membrane with channels which can move substances. The cellulose in the cell wall, obviously it has got the key features that you need to be able to describe. So if they're asking about movement of water, you should know about your symplast pathway, you should know about your apoplast pathway. If it's going through the apoplast pathway, that is the cell wall and intercellular space. It's going through the cellulose fibers because they are allowing the water to flow freely through it, and the water obviously has the cohesive properties. The symplast pathway is going across the membrane, so through the symplast pathway it has to get into the cytoplasm, and then it's going to make its way through the plasmodesmata until it gets to the xylem. Peptide hormones are not the same as non-steroid hormones. Non-steroid hormones can cross the phospholipid bilayer, whereas the peptide hormones will have to bind to a receptor on the surface. With regards to how the plants have specialized, this is one of that module 2 differentiation. I do feel like this is coming up. I feel like they're going to ask you potentially about that. A concentration inside water potential. If I said to create a concentration gradient, no. Be careful, Ninja, be careful on that one. If you were saying to create a solute concentration gradient, or if you're talking about water, you should always talk about water potential. So we're going to create a concentration gradient using ions, using mineral ions, using solutes. Therefore, that will affect the water potential, and therefore water will move down the water potential gradient. So try to be specific on that one. Now the xylem vessel, obviously dead, hollow, will actually, it started off as being a cell which was produced through the process of mitosis. So we know we have our meristem cells that will go through mitosis, dividing, We knew, because we saw it earlier, cytokinesis can occur, and therefore if we're making more cells, that will therefore need to go through that differentiation process. Are they going to differentiate into the xylem, or are they going to differentiate into the phloem? Because if we're in the cambium in the middle, that differentiation depends on which side it's being pushed out of. With the xylem, that primary cell wall is made up of cellulose. A secondary cell wall is going to be made of lignin, and that can be in lots of different patterns depending on the xylem vessels, because remember, it's not just one, it's lots of them, and that allows for them to have these different functions. So if it is spiraling, that allows the flexibility. If it is the pitted ones, that allows the lateral flow of the water. If it is the ring-like structure, that's going to prevent the collapsing. So each of the different patterns is important for the role in which the xylem needs to take, because remember, the plant wants to grow upright so it can collect as much of the light for photosynthesis. So it needs a structural support, and that's where the lignin is important. Now, if we're going to dissect the plants, if we're wanting to observe the movement of the liquid through, we can either do a transverse or a longitudinal section. And in this case, with the daffodils, what we did is we split the daffodil stem up. We put half in a dye and half just in distilled water. I tried to repeat the same thing a number of years ago in my living room. You can see the little playpen in the background, but it took so long. I didn't realize it was going to actually be so long. And so therefore, I'm back to using my nice little pictures. And so if we're looking at the observations, we would use a stain that would move up. So we need to look at water property. A non-steroid hormone and non-lipid soluble. So if it's a non-steroid, hold on, did you say, oh sorry, you said peptide or non-steroid? Yes, you did. I thought you said, I read that, I misread that and read it as a steroid hormone. I apologize, you're right. Peptide hormones are non-steroid hormones, and so therefore I was answering it as if it was a steroid hormone. I apologize, you are right on that one. Water, if we know the structure of water, we know we have our polar molecule, we know it's uneven sharing, we've got two hydroxyl groups, and therefore we can have hydrogen bonds form between our water molecules. Cohesive property, therefore, and because of that, when we look at liquid at room temperature, there's so many hydrogen bonds, it means that they are collectively strong, which is why aquatic organisms are able to live in the aquatic environment, because there is a high specific heat capacity, which means that is a stable environment. So that's where they could bring in ectotherms and fish in aquatic environments. Now, if they're looking at cooling and endotherms, the high latent heat of vaporization, lots of energy needed to break the bonds. If they want to look at the fact that ice is less dense than water, then they could look at the high latent heat of fusion, and you would need to know about that ice being less dense is going to float on the surface. Therefore, it's going to create a layer, which is an insulation layer, and therefore the environment underneath is able to be stable as well. The adhesive properties is the attraction of water to something else. So whether that attraction is to, in this case, this stick, or whether that attraction is to the cellulose in the xylem vessel. One thing that I really love about this is you can actually see if, in this case, the waterproofing properties of the leaf means that these droplets can be separated, and when I allow them to come close enough together, these two big droplets, you'll see them cohere together, and that's the cohesive property. And then you'll see in a minute that they're going to flow as if they're one, and they flow as a stream as if they're one, and that's the transpirational stream. So when we look at the effect of cohesion and adhesion, that results in what's known as the capillary action, and that allows the water to move up through the xylem, and it moves up against the force of gravity. And so the other things to note going back is water, solvent property. It can dissolve all these different cations, anions. The fact that if we're talking about the plants, it can therefore carry in the apoplast pathway and in the symplast pathway. In the xylem is the transpiration stream. In the phloem, it's translocation. In plants, it's also allowing those hormones to move. And obviously in animals, it's allowing the hormones to move because they're going in the blood plasma in the animals. They're also, with the solvent property, the fact that it's a medium for metabolic reactions. The cytoplasm, the stroma, the mitochondrial matrix, these are all liquid-filled solutions which are going to allow for these metabolic reactions. If we're looking at the kidneys and our nitrogenous waste, is a way in which we can dilute toxic substances until we are able to excrete them. Obviously, you've got your surface tension. We've talked about surface tension already with regards to our lungs. And the surface tension allows us to get these water droplets, and then that water can run off and go into the soil. With some organisms, for example, these pond skaters, they can actually live on the surface of the water due to that surface tension. As long as they are not breaking the hydrogen bonds, they are therefore able to skate along the surface. And obviously water being transparent means light can get through for those aquatic plants which are going to be photosynthesizing. But be aware that when the light that comes from the sun enters into a different medium from the air into water, it is going to refract, and that will disperse it, and you will end up with different wavelengths hitting different points. And that was a question that came up when it looked at photosynthesis before. Whew, okay, we're going to get there. Okay, transpiration then. Transpiration, obviously loss of water. It's going to happen because we need the stomata to open. We need carbon dioxide for the Calvin cycle. We know osmosis is going to bring the water in through the root hair cells into the symplast pathway, into the xylem vessel. We know we've got cohesion and adhesion. We therefore know we've got our transpiration stream. So if we understand that, if we understand that transpiration is going to happen, we need to be aware that we have different organisms. Some that live in areas where there's not very much water, and others where there's lots of water. So if we have a xerophyte obviously adapted to the environment where there's lacking water, they're going to reduce the surface area of the plant, reducing their leaves, rolling their leaves, having these spines. They're going to have storage of the water in stems. I could see a question coming up looking at maybe an animal against a xerophyte and how they have adapted to survive in areas where there's not very much water available, for example. So we've got thick waxy cuticles, and obviously stomata number is going to be reduced or sunken or surrounded by hairs, and only opening when transpiration rate is lowest. If they're hydrophyte, they're submerged in water, they don't really have to worry about this. Because they don't have to worry about it very much, they're therefore going to only have some adaptations which are related to the environment for which they live in. Okay, now the phloem, one of the key things that keeps coming through, and it threw a lot of students off last year, was the phloem sieve tube element is the name of the cell that makes up the phloem tissue. The phloem is the tissue which is inside the organ, which is the vascular bundle, which is in the stem or in the leaves or in the roots. So it's important that you know you've got your phloem tissue, your xylem tissue, and you should know how to identify where the xylem and phloem is, if it's in the stem, where it is, if it's in the leaf, and where it is, if it's in the root. So if they do ask you, going back to the calibration of a graticule, can you actually use this to work out? So if the diameter of that vascular bundle is 13 divisions, and I tell you that each of the divisions is 25 micrometers, you should be able to then work that out. How have animals been adapted for a lack of water? So you would expect an animal that lives in an area that is very dry to have a longer lip of Henle. A longer lip of Henle means that they can reabsorb more water back into the bloodstream and therefore survive a longer period of time where they don't require that intake of water. Also, you would expect an animal that maybe has to hibernate over the winter months, again, to have a way in which they are reabsorbing the water so they're not losing as much water potentially. If we're looking at translocation, we need to know some key things from our source to our sinks. We need to know about loading. Is it the apoplast pathway? Is it the symplast pathway? We need to know about mass flow. We need to know that by loading our assimilate, now don't think it's going to be about sucrose, it could be about amino acids, just to be aware. They could throw a little spanner in the works by telling you that it's a amino acid that's being translocated. If they do, then that amino acid being translocated is the same way as doing the sucrose, and it should say somewhere that it happens in the same way. So we're going to load it. If we're loading, we're therefore affecting the water potential. We're therefore going to have water move in by osmosis, and that's going to create a hydrostatic pressure, and that water moving in by osmosis will be lateral movement of water. And so if I just add that in here, we know that our water moves up through the xylem, and therefore it is going to move laterally from the cells and from the xylem into the phloem sieve tube element, wherever the source companion cell is. And therefore we are going to then have that being translocated to the sink, where it's going to be unloaded, where it can diffuse, it can then be converted, and then that will again change the water potential. Now be aware that your, for example, tubers could become the source, and that translocation would be done in the opposite direction. If we're going from the source, it could be going up to the new growing leaves above. If it is where it's the source is the seeds, when the seeds germinate, they become the source. So it's important to look at where the question actually is, or what they've told you in the question. Okay, photosynthesis then. We know photosynthesis happens in the chloroplast. We know that the chloroplast has got its thylakoid. We know that the thylakoid is a flattened sac, and in the thylakoid we know we've got our photosystems. We know those photosystems are going to absorb a photon of light. When they do, that is going to allow for the electrons to be transported through the electron transport chain. So whether we're talking about this or whether we're talking about respiration, we need to always say about the electron transport chain. Whole point is about these hydrogen ions. I'm going to quickly skim through this because I feel like you're not going to get a lot on this one because it was done loads in last year's exam. But we're going to have hydrogen ions where they're going to move through, and you obviously have the production of ATP. The reduced NADP and the ATP are then going to be used in the Calvin cycle. Now, if I have my electrons, because my photon of light has gone and my electrons have gone from photosystem 2 to photosystem 1 and photosystem 1 to reduce NADP, I need to replace those electrons, and therefore to replace the electrons into photosystem 2, I need photolysis. I need to break the water down, which releases the oxygen. And so therefore it is non-cyclic photophosphorylation because there is no cycling of the electrons, whereas in cyclic photophosphorylation, we're going to only use photosystem 1, and because we're only using photosystem 1, we are therefore cycling the electrons back through the electron transport chain from photosystem 1 to photosystem 1, and therefore that is going to allow for the hydrogen ions, and allow for chemiosmosis, and allow for the ATP production. The light independent reaction is going to therefore use those products, and it's going to use those products to fix the carbon dioxide. Carbon dioxide then is going to break down into GP. That is then going to be reduced into triose phosphate. One triose phosphate can go off, and the rest are going to be regenerated back up into the ribulose biphosphate. So if they're going to ask a question on that, I don't really feel like it's going to be lots on the process because they've done so much of it lately. So I would say it's not coming up, but I'm not feeling like it's going to be a big one for paper 1. So I would say don't not look over it, but just remind yourself about it. If we're going to look at the triose phosphate and where it could be used, the triose phosphate can obviously be used to produce glucose. Glucose can be used in respiration. The triose phosphate can be used to produce the fructose. The fructose and glucose can be used to make the sucrose. The triose phosphate can be used to produce the cellulose. It can be used to produce the starch. So starch we know is a polysaccharide, and therefore we should know it's made up of amylose and amylopectin. We should know that we have an iodine test that we would use and what the negative and positive results are to show that the starch is present or not. Now, if they are going to go down the route of plant response, I'm trying really hard to give you as much as I can here. I do feel like ethene would be coming through if they're going to do plant response. Could be about leaf fall. In autumn, light levels drop. Decrease in oxygen concentration is going to increase the production of ethene. Ethene is then going to affect cells close to the region that it's going to be produced, and it's going to switch genes on. Those genes are going to produce enzymes. So the ethene is going to activate protein synthesis occurring inside the cell, and that is therefore going to end up with the digestion, and that will therefore cause the leaves to fall off. And we can see the different photosynthetic pigments in our leaves because we can see the yellows, we can see the reds, we can see the orange, the accessory pigments. The other thing that ethene can do is ripen our fruit. So decrease in oxygen concentration. Again, that decrease in oxygen concentration actually comes from the fruit. When the seeds inside the fruit have matured enough, they will then signal for a decrease in oxygen concentration, which will then increase the ethene concentration, and that will therefore stimulate the fruit ripening, which is a biochemical change inside. Now, I feel like they're more going to look at the commercial use of the hormones. So we can promote fruit ripening if we harvest the fruits early. So we can do it under controlled conditions. We can promote fruit to drop by increasing the ethene concentration. One of the key things to note is they're not going to not talk about other hormones. They did a lot in gibberellins last time, but gibberellins can actually be used to decrease the ripening process. Now, we talked about transpiration. We've talked about the transpiration stream. What you should know is stomata being open or closed is actually dependent on the inorganic ions that are moved. So if the potassium ions are actively moved into the guard cell, that will increase the solute concentration, and that will therefore result in a lowering of water potential. Water will enter by osmosis. If we do the opposite, if we actively pump the ions out and water leaves, that will cause the cells to become flaccid, and that will close the stomata. Now, I feel like they could link this to abscisic acid. If they are in a region where there is water stress, ABA can actually be produced by leaf cells if there is either too hot temperatures or if there is a shortage in water availability. So the ABA will bind to receptors. Remember, if we're talking about any type of hormone, the hormone will bind to the receptor. Where is that receptor? And if the receptor is on the surface, then it means that the hormone can't get in. If the receptor is inside the cytoplasm or it's in the nucleus, then the hormone can get in. So use that as your guide. By the ABA binding, that is going to activate changes in the ionic concentration. That will therefore change the water potential, therefore reduce the turgor, closing the stomata. If there is water stress, if it's in drought, then the ABA can also come from the roots, and that will therefore mean that it will come all the way up through the xylem, again still binding to receptors on those guard cells, because the guard cells are the ones which are differentiated for the closing. Yeah, it did come, Zara, you're absolutely right. Gibberellin came up a lot last year, but it doesn't mean they're not going to do plant response. It just means how they assess it. It just depends. I feel like if they're going to do it, ethene and abscisic acid are going to come up more likely than gibberellin and auxin, but it doesn't mean it won't come up. So you need to make sure you are confident in all the topic areas. But I would say they're more likely to go on commercial use of the others. Another thing to note, if we're changing the ion concentration inside the cell by abscisic acid, it can also be used as an antifreeze molecule, so in winter. So by adding solutes into the cytoplasm, that's therefore going to lower the freezing point, and therefore that is going to prevent the chemical reactions from completely stopping. So the other thing to know is they could ask about auxins, they could ask about phototropisms, they could ask about apical dominance, and if they do, then make sure that you are talking about where there is a higher concentration and whether that higher concentration is going to increase the cell elongation, or whether it's going to inhibit the growth. And if it's inhibiting the growth, then that would be showing you apical dominance. So if we're going to commercially use auxin, how are we doing it? Are we using root powders? Are we using it with our micropropagation? In that case, that would be a paper 2 question. Are we using it as a weed killer to prevent competition? Are we using it as a way of producing seedless fruits? Obviously, like grapes, for example. I love a seedless grape. I don't like seeded fruits. And therefore, that could be a use of, a commercial use of the auxin. It could be about gravitropism, if it is about gravitropism, but again, these came up in the last couple of years. If it does come up on gravitropism, again, it's to do with that concentration. Again, it would be a question where you will need to pull the information out of it. And the only other thing they could talk about is about plant response to herbivory, and that plant response to herbivory would be obviously where the plants are producing their spines, their thorns, their spiky bits, or they could be producing the chemicals. Now, if it is about the Mimosa pudica, the one which is responding to touch, that is going to reduce the surface area of the leaf. Again, that one's to do with moving ions in different directions, and that would therefore prevent the herbivore from eating, because the plant seems damaged already, it's already been eaten, and then it will move the ion concentration to open the leaves up again. And then you've got obviously your chemical substances that they could make, like caffeine, etc. I could see them potentially doing a question on aphids tapping into the phloem to feed on the sap, and looking at translocation with phloems. They had a very small question on callos in last year's paper, looking at the sieve tube, the sieve plates being blocked up in callows. Now that's more paper 2, but it could come in with a question on translocation, because not that it's for your exam board, but in previous exam boards they have done questions where they have the aphids have tapped in, aphids, but they cut off the stylet of the aphid, and therefore they measure the rate of the flow. So they're measuring the rate of translocation through that process. They could do something along those lines, but I don't know. Guys, I am going to stop there because I have been going for a good 4 hours of teaching now. I've tried my best to cover as much as I can of the content. I think I've pretty much covered everything. If I haven't covered anything specific, then obviously go and check the playlist for that module area, because you can still see the videos that I've got on it. I hope that's helpful. Good luck to everybody for Thursday. I won't be available tomorrow because I have my clients that I will be tutoring in preparation tomorrow. So good luck, everybody. Thank you so much for coming and watching and liking and sharing. I do appreciate that because that makes a big difference for me. The more people who know of my channel, the more people who comment, like, share, etc., the more they push my stuff out. So I do appreciate it. So, oh wow, thank you. Just noticed I got 2,000 hearts on TikTok. Thank you so much. If you have been watching on TikTok and you want to actually look at the screens and the slides that I've been doing, then please do jump over to my YouTube channel. It will be in the live. There's 2 videos. For those of you who were in your chemistry exam this morning, the session 1, the 2 hours I did this morning, that is available as well. I am now going to turn these into a PDF, and I will upload that for you. If you are interested in purchasing these notes, then I will make them available on my website, which is ukbiotutor.com. I'll do that just now, and thank you, because anyone who does purchase them, obviously that helps me to be able to create more moving forward, and obviously this will still be helpful for you for paper 3 as well, because if they don't ask the questions in paper 1, they can still ask it again in paper 3. So although the module 1, 2, 3 and 5 is in paper 1, if they don't assess it paper 1, those kind of topic areas could still come up in paper 3. It just depends. So guys, thank you so much. Have a good rest of your day, and I will try and do another one of these next week. I've not got as much time next week as I've had today, but I will try and do the same sort of thing as best I can. So thank you and goodbye.